Hello, everyone. Thank you so much for coming out for the seminar. Really excited to have two friends, one of whom is giving a talk. I'm really excited to uh, introduce Aniruddha Belsare. Dr. Belsare got his degree in Bachelor's of Veterinary Science from Mumbai Veterinary College. And then he was one of the few wildlife vets in India for a long time, which meant that he's worked on king cobras, leopards, crocodiles, bears, elephants, and a bunch of other just animals. Because whenever people thought of, oh, we need a wildlife vet, oh, I know Anirudh, let's call him. But then he sort of left active veterinary science to come when he came to the US and did a PhD from the University of Missouri, and then has also again over the last decade has worked in several places in the U.S., including Missouri, Idaho, Montana, and one more place I forget. But yeah, Anirudh has really wide interests, and I'll let him do most of the talking. So without further ado, welcome Anirudh.
Thank you so much for the introduction. I'm super excited to be here and interact and talk with the vibrant crowd of researchers. Um, I already started talking some of this stuff since morning. I have met several of you, and I am going to talk about the wildlife disease systems, investigating wildlife disease systems, mainly as in the wake of COVID 19 pandemic, wildlife diseases have come to the fore again. It's not the first time, right? And just in the first quarter of 21st century, we have had several outbreaks. Uh, SARS Corona 2003, uh, we had swine flu, MERS, which is the Middle East Respiratory Syndrome, ongoing in the Middle East region, several Ebola outbreaks. We have had Marburg, then, of course, COVID-19 and Mpox. And I can just keep naming them. All of these are in many ways wildlife associated diseases, right? And what it means is there is a critical need to better understand the epidemiology, the ecology, the transmission dynamics of these diseases and similar systems. Not all of the diseases that I mentioned were new, but some of them were in new regions, new areas, and looks like that is what we are going to deal with in the coming years. So we manage or we have to manage diseases that have public health implications or conservation implications, and of course, if they have economic implications. With that, I just want to say I'm going to focus on three of the disease systems that I work on. These will not necessarily cover all of the important aspects of the approach I want to discuss, but I think these are kind of relevant to the work that you do, to the wildlife that you encounter. And that's the reason I chose these three. I'll talk more about it soon. But no disease ecologist worth his salt will ever not have an epidemiology triangle in the presentation. So I'm rather going to start with that. What I really want to emphasize here is that, oftentimes, including healthcare professionals, including several scientists, researchers in the field, disease is seen as uh, a host meets a pathogen, a susceptible host interacts or meets with a pathogen, and disease happens. That is a very simplistic and often a wrong understanding of the disease system. And therefore, we came up with the disease ecology triad, which is these host pathogen interactions occur in an environment. right? And all of these are connected, but this does not necessarily capture all the complexity. These are inherently complex systems, especially when wildlife species or free living species are involved. The complexities increase. And one of the reasons is that most wildlife pathogens are what we call as host generalists. They can infect more than one species of host. And zoonotic diseases, by definition, have to be multi-host pathogens, right? So there has to be multiple hosts interacting, resulting into an increased complexity. All these interactions result in complexity. Also, not all hosts are the same, right? They do not really uh, have the same impact or transmit the disease equally. And so there is host heterogeneity include. Furthermore, in any real world system, you cannot really expect that a host, even us humans, to be infected with only one pathogen at a time, right? It's sometimes helminths, worms, parasites, pathogens, and there are multiple things interacting within a host. So that adds to the complications. There is host evolution, pathogen evolution, pathogen biology. Then you might not see that here, but then in some cases we have vectors involved in a disease system. So vector biology and the range expansion of these vectors, all of this together results into a system with many interconnected components, right? And these interconnections are what uh, is what really responsible for the challenges that we have to kind of deal with in managing such systems. So, of course, I don't want to say this out loud, but there are other components as well. Um, with wildlife diseases, with pathogens of wildlife species, one important point to keep in mind is the hosts move freely, unlike companion animals or humans or their uh, livestock. Right, the hosts move freely. Their pathogens also move freely. So that is an additional layer of complexity. So these hosts are often difficult to access for surveillance, for sampling, or even for treatment, let's say. There therefore are limited tools available to us to actually mitigate the risks posed by wildlife diseases. And also, please keep in mind that with wildlife diseases, if at all there is any treatment or if there is any management intervention, it is applied at a population level, not at an individual level. So these are all different layers of complexity. One important issue here is this is a complex system. There are a lot of components, and any perturbation to such system results into a non-linear outcome. This is something which, again, is difficult to explain to a kind of a lay audience, but I'm sure you will understand. So complex systems, you change something, it does not result into an outcome which can be predicted. What you actually see here, and I'm going to bring in an example that I am super impressed with in the next slide, but we have non-linear outcomes, which often means that your remedies can be more dangerous than the maladies itself. That's a huge issue with wildlife diseases. And we have seen several examples. I don't want to waste time on that. But as I said, I do want to uh, talk about not considering these complexities and uncertainties. If you don't consider them, you might end up with what is called as tilting at the windmills or Don Quixote. So quixotic response is what we have often done historically. And to not do that, we need to take all of this complexity into consideration. And so this is where I want to talk about this really intriguing study done by Ezenwa and Jolles. And this is one such study where I really feel envious. Why am I not a part of this study, right? I think this is a seminal work done in the recent times. So these folks were looking at populations of African buffaloes, right? Now, these populations are naturally exposed to bovine tuberculosis. So, bovine tuberculosis is a bacterial disease. It's a disease of huge economic importance for cattle and for wildlife. So, these populations are naturally exposed to BTB, also naturally exposed to helminths, that is, parasites, roundworms. So, what they found is there, based on their study, they found that there is a negative correlation, as you can see on that plot in the middle, between prevalence of tuberculosis and prevalence of helminths. So on the x-axis, you have worm prevalence, which is here you have the worm prevalence, and this is the TB prevalence. So there is a negative correlation. What that means is herds that have a high, TB prevalence have really less or no worms, or within herds that have you can actually look in herds, and uh, animals that are TB positive are usually worm negative, and animals that are worm positive have a very lower prevalence of tuberculosis. So this negative correlation was known. This is not surprising, because we know that helminth induced immune suppression actually results into a more susceptibility, more vulnerability to bacterial and viral diseases. So the body's immune system has to deal at the same time with the parasites and with the viral or bacterial infection. So if there are parasites, the body's ability to deal with TB goes down, and the outcome is kind of negative. So the obvious question then arises, because we want to conserve wildlife, and TB is a disease that we introduced into the wildlife, so we need to deal with it. So one logical way would be, so before I go there, what this data also tells us, if you look at this data, there are hardly any points here which have high TB prevalence and high worm prevalence. Why is that? Because animals that are co-infected die. So you don't have any points here, right? Because of co-infection and the effect of, the co-infection. So the point is, can we then go in and deworm African buffaloes that are co-infected and improve the outcome of these co-infected animals? So like we give deworming medication to dogs and cats that we have as pets. Can we go in and give a dose of deworming to African buffaloes that have both TB and worms? So now the worms go away, and maybe the animals would have a positive outcome despite having tuberculosis. Makes sense, right? So they went in and did that. What they found was, yes, indeed, after treating the co-infected animals with deworming, the individual health outcome improved. Uh, Buffaloes that were co-infected had both BTB and worms. Now the worms are gone. They lived longer. So individual outcome improved. But at the population scale, what happened, unfortunately, you cannot see it because of this thing, but I'll explain what it is. At the population scale, because infected, TB infected animals were living longer, they actually were spreading TB much longer than without the treatment. So you go in and treat the animals with good intention, that actually leads to hell, because now you have more TB in the landscape. So this is what we call unintended consequences. This is also a non-linear response by the system, right? So you would expect a linear response from a simple system: give deworming, improve the chances, and all is well. That doesn't happen, right? And the reason I'm fascinated with this study is because this is an analytical approach used by the authors. Field experiments. We see very few studies like this. We have been talking about this since morning. We see very few field experiments nowadays, right? Also, what I really want to point out um, is that the analytical approach need not always be model-based approach. And for a long time, after the discovery of, or after the germ theory of disease was proposed in late 19th century, much of our research, much of our work on diseases has been really descriptional. Like, this pathogen causes this disease. These are the antibiotics you use for the disease. This is the culture. This is the diagnosis and all of that. So that's a descriptive study. But since 1980s, late in the 20th century, this approach changed. And we now are really doing analytical studies of diseases. And that analytical study, this is one example. The other example is what I'm going to talk about today, which is like a virtual experimentation, right? And in no ways I am suggesting or saying that virtual experimentation can replace field experimentation. There is no replacement of field work or field studies, right? And that's what I'm going to talk about for the next two slides, that as these are complex systems, right? As these are systems which have non-linear responses, right? We need to really use a triangulation approach wherein we have to gather data using multiple methodologies which are current and relevant. Get that data, put that data into a kind of a analytical framework, and then answer the questions we have. In formulating these questions, again, for most wildlife diseases, stakeholders play a very important role, and they need to be a part of this process, especially of building up the questions that we want to ask for, or deal with such systems. Now, the analytical approach, as I said, could be field experimentation, but it could also be virtual experimentation using models or using analytical framework. And what it does is it integrates data, integrates observational, experimental data together in a formal framework. This can help us, at the very least, prioritize our research, the questions that we need to ask, the data that we need to better understand these systems so we can effectively mitigate the disease threat. What it also does is helps us elucidate the complex dynamics, deal with the uncertainties of the system, and again, mitigate it effectively. And finally, from a very application point of view, this helps us run these virtual interventions, virtual strategies to compare and contrast alternative strategies. What will work? What will not work? Right? Rather than going in the field and doing field experiments which are expensive, which take time. So the context here is if we have an outbreak of some new disease, are we going to wait for 10 years to better understand that system and then come up with interventions, compare A and B and C and say, okay, after 10 years, I know A works the best? No, we really need a tool wherein with all these uncertainties, with the new and emerging diseases, we can, test drive interventions and come up with the efficient way of mitigating the threats posed by such systems. So just want to reiterate, the analytical approach I am going to talk about, the modeling approach, does not replace field work, does not replace field experimentation. It actually complements, right? So the whole purpose of this virtual explorations is to find the Achilles heel of this complex system so that we can manipulate the system and have better outcomes despite the uncertainty, despite the complexity. Like that is the goal with virtual model-based explorations. And in this kind of modeling framework, I use agent-based approach, agent-based modeling. There are many other modeling approaches. There is a reason why I think this works best for disease systems. And it does not mean that this is the only way to do modeling. I do this especially with infectious diseases. Is I have a very simple explanation for it, and not everyone will agree with it. But um, for any infectious disease, right, which has an agent that causes the disease in the susceptible host, whether it's a bacteria or a fungi or a virus or a protozoa, whatever, the susceptible host has to come in contact with the infectious agent. That is how infectious diseases are transmitted. So this could be coming in contact with another infectious host. or it could mean coming in contact with the environment that has these infectious agents, right? But the individual, the susceptible individual, has to come in contact with the pathogen. And this is driven by very individual level processes. This is an individual level process. If I am a white-tailed deer and I'm a part of, like, let's say, a bachelor group of, like, bucks, then I interact with group members at a certain frequency. I do not interact with the dose social group all throughout the year, except when, it's the rutting period, right? Or there is a feeder somewhere in the landscape which I go to, and that's where other deer also come from other groups. So this uh, is kind of driven by the contact network, which is based on the behavior and the biology of the species. But all of this is an individual level process. And in an agent-based model, that is what we do. We simulate individuals in the population of interest, whether it's one species or multiple species, as agents in a hypothetical landscape. And these agents interact with each other, they interact with the environment. And these local interactions result into system-wide patterns. And that is what we are interested in. So in many ways, the agents are endowed with real world attributes, like age and sex and behavior and several other things, immune status. They have these, and the rules of interactions are based on these real world attributes. So agent based models can incorporate a lot of heterogeneity, right? There are other ways of modeling diseases, which are like, as we talked about, compartmental models, SIR type models, right? Which treat individuals in the population, they kind of puts them into compartments. All of these are susceptible. All of these are infected. All of these are recovered. So there is no heterogeneity in those approaches. But here we can incorporate all the heterogeneity. We can incorporate a lot of uncertainty, and yet have this, simulation that we can explore. So that's the reason I use and work with individual based or agent based models. They are spatially explicit. I can incorporate real world landscapes into my models. And so my agents, the species or the hosts that I am simulating, can actually interact with the landscape. There is a realism there. So with that, I do want to get on with the first case study that, I had the opportunity to work on, and wherein we developed an ABM, which is an agent-based model, to explore the effects of individual host infectiousness on population level transmission. And let's talk more about it. So this is about the West Nile virus, right? Up until um, up until 1999, there was no West Nile virus in the Western Hemisphere, right? It was one occasion of introduction in the summer, May of 1999, that this particular pathogen was introduced in New York City, that star over there. And the reason I put so much emphasis on it, this is kind of a reminder what an emerging disease, the cost of an emerging disease, not only conservation or public health. Overall, West Nile virus is now the most common arboviral disease in the US. It's been 26 years now since introduction, right? And millions of birds have died, continue to die. Millions of people, not millions, thousands and thousands of people suffer from neuro-invasive West Nile virus. All of this was not really there before 1999. And that's what we are going to talk about in this first case study, right? So 1999, this was introduced, and very rapidly, it spread through the contiguous US. Within like 2 years, it came here to Florida. And in Florida, this because mosquitoes are involved in this, and the environment here, it became enzootic. By about 2001, West Nile virus was enzootic in the mosquitoes of Florida, right? And it rapidly spread to, it had this westward expansion. We believe it's because of the migration or the migratory birds, right? So Florida is important in this story, and I thought this would be a great case study to discuss with you guys. So, that westward extension of West Nile virus, within 2 years, it was in California, and millions of birds had died all across the US. It's also thought to be because of this elliptical migration route, like birds coming down on the eastern seaboard, but while going back northwards, they use that internal routes. And then that's how the virus spread to the local mosquito populations and then to the west of the US. Um, as I said, it's currently the most common infection, parvoviral infection in US. These numbers are from 2024. I don't know if we have any updates currently on the CDC side, but uh, 49 serious human cases in 2024, again reported, 1,000 neurodegenerative cases in 2024, and I cannot see that, but total human diseases reported. And you can see almost all states reported West Nile virus in 2024. And the state that hasn't reported does not mean it was not there. And I'll tell you the reason later. Um, right. So West Nile in nature is maintained in a cycle between birds and culex species of mosquito. And all birds in the US are susceptible to West Nile virus. Some will show overt signs of disease and die, like house sparrows or crows. Others might get the infection, but not really show any signs of disease and can act as amplifiers or super spreaders of the disease, right? Humans can get it, but humans are what we call dead end hosts. So humans get it, and they do not have sufficient viremia to transmit the disease back to mosquito. Similarly, horses can get it, but they cannot have sufficient viremia to transmit it back to the mosquito. So humans, horses, dead-end hosts, and maintained in birds and culex species of mosquitoes. Again, back to this disease epidemiology, triangle. American robin, largely migratory bird in North America, right? We think of this as a super spreader. You know of typhoid Mary, right? So this is like the typhoid Mary, not because it has a high viremia after infection. It actually has a moderate viremia once it gets infected. Doesn't die of the disease. For a few days, maybe 2 days, it gets like slightly high level of viremia, and the viremia goes back down, and then it's okay, right? But Culex species prefer this as a source of their meal, this bird. So they have a lot of blood meals from American robins, and that's why this goes into the super spreader category. Unlike the other fellow, which is the northern cardinal, this fellow cannot really get that level of viremia. So it can get infected, but it cannot generate sufficient viremia to pass it on to mosquito. So this is the super suppressor, and this one, the robin, is the super spreader. And we can look at other species, and they will fall in between or be super spreaders or super suppressors. This is important to understand the complexities of the transmission or the transmission dynamics. So we'll focus on American robin. And in mid-Michigan, there is this bird banding station. Michigan State University runs it, and Dr. Owen and her colleagues, they have like been banding birds for several years, and over the years they collected a lot of migration phenology data and the number of robins coming to the station, leaving the station, what days, and the age composition, the age ratio, the the numbers and dates, and all of that data is available, was available with them. They also did experimental work, uh, looking at, like, took the robins in the lab and did some experimental work wherein they looked at the effect of stress, mostly food stress, on American robins, and how do these robins respond to West Nile virus infection if they are stressed. With the migration, they often, with the change in the or the human anthropogenic changes in the landscape, this is thought to be an important issue, food stress. So their experiments with food stress, what it revealed is food stressed robins have a higher level of viremia for a longer duration. So the red ones are the non-food stressed robins. You see, they get infected. There is a spike in viremia, but it rapidly falls down. But with food stress, you have a higher level of viremia, and the viremia persists for a longer duration compared to the non-food stressed birds, and then it falls down. They don't die of West Nile virus, right? So there is a slight change in the individual response to West Nile virus, depending upon whether they have food stress or not. Does that make sense? Right. So this is the experimental data. We have all the bird migration, phenology, and all of that data. And the experimental data tells us that the infectious index for food stressed animals or birds is pretty high. Now, the question we then asked is, what does this mean overall for population level transmission? What does it mean for the public health risk of West Nile virus? We have this data. We have this cool experimental data. How does it translate? So because I was involved, I came up with an agent-based model of this system. Now, this is a hypothetical landscape. The published model is over there. The square that you see, we have a hypothetical landscape, and we have uh, American robins and mosquitoes as individual agents simulated on this landscape. Now, the number and all these processes that are important for West Nile virus transmission, the stochastic processes, they are all incorporated in this model, right? So we have mosquito population dynamics, we have mosquito robin interactions or the biting rates, we have the arrival of robins, the dates that they leave, the time that they leave. So essentially, the model runs from August 1 to October 31st, a 3-month period when robins are actively migrating in mid-Michigan, right? And then we have all the phenology incorporated in there. And the questions we were going to, or we asked of this model is, what's going to happen if we have more food stressed robins in the population? And that's what you see here. The baseline is red, right? And then you have 10% population stress, 25%, and 50%. And we can get as nuanced as we want to because it is a model and we have full control on what scenarios or what parameters we can adjust. On the y-axis, we have mosquito infection rate, right? And it is per 1,000 mosquitoes. So essentially, prevalence in mosquito. So how does food stress in robins kind of correspond with the infection rate in mosquitoes in the region, right? As you can see, even slightly food stressed robins, only 10% of robins in the population are food stressed, the mosquito infection rate significantly increases, especially in the period when a lot of robins are migrating. So which is mid-September to kind of mid of October. That's when you have a lot of you have a increased mosquito infection rates, and especially pay attention to uh, uh, this level of MIR. This corresponds with real world human outbreaks. So we are not predicting that food stress at this level will result into human outbreaks. But what we are saying is we do find a definite impact of food stress in the robin population on the mosquito infection rate. And this mosquito infection rate corresponds well with human spillover risk. Uh, Although all of this is based on data for mid-Michigan, the model is flexible, so we can use it for any other areas, and we can parameterize it with data from areas of interest to essentially forecast the impact or the public health risk in such situations, right? So I just wanted to use this case study to highlight how we can bring together observational and experimental data to kind of better understand the working of such complex systems, right? So food scarcity results into change in the within host effects, essentially increased infectivity in the robin, and then high prevalence of infection in the mosquitoes, which can result into a super spreading event. That is kind of the elucidation of the dynamics in here. The paper is available online. I think it's an open access paper. With that, I do want to get on to the second case study, which is again raccoons. I believe they are everywhere, so it should be of some interest here as well. And now instead of a virus, this is an intestinal roundworm, Baileys ascaris procyonis. Right? It's a very interesting roundworm. Again, that's a morbid kind of statement, but the reason is because it's a very typical roundworm for a raccoon. It lives in the small intestine of the raccoon, doesn't do anything to the raccoon, which is a definitive host. Like they just harbor the worms. But the worms, the females, are prolific egg producers. So look at the numbers. 15,000 to 77,000 ova per day per female worms thrown out in the feces every day. And the peculiarity here is these eggs are extremely hardy. You just cannot destroy them. If you have a raccoon latrine anywhere on the campus and are worried about bee pro, you have to take a torch and burn it. Nothing else works. And that's what the pest control or those agencies do. You have to burn it. No amount of bleach or any other stuff, but the eggs are very hardy. They can remain infectious in the environment for multiple years. And the real worrisome thing is these eggs can infect the raccoons and complete the life cycle, but they also are so generalist, they can also infect several vertebrate hosts, including humans. And in humans, and the other hosts, which we call paratenic hosts, because they are not required for the life cycle of the worm. But in these hosts, they just insist. And then when the raccoon consumes that mouse or whatever, they kind of come out of the cyst and infect the raccoon and complete their life cycle. With humans, these eggs, we being the dead end host, the eggs, the larvae get lost, and then they can end up into the retina, causing ocular larva migrans, or in some of the organs, causing visceral larva migrans, or in the brain, causing neural larva migrans. And most of the cases documented so far have been in toddlers, kids young than 2 years of age, and it causes a really bad neurological disease which has no cure. And we think these cases are rare, but they could also be underreported. Also keep in mind that there are several reports of human Baileys ascariasis, and they are increasing, more reported more and more, but still not sufficient to cause any policymaker, bring it to the attention of policymakers. So they still are thought of as rare. One point I always make with such rare diseases is it's like Russian roulette. There are 999 empty chambers, but only one chamber has the bullet. But the consequence of encountering that one chamber are very high. The cost is very high for the individual who gets it, the family members, even the community, right? So anything rare is not necessarily zero. It's non-zero. Can happen, it will happen sometimes. And when it happens, it's really bad. So this is one of those diseases. Raccoons are everywhere. The reason I wanted to talk about this case study especially is the map that you see here. And this is like that they are talking about uh, the prevalence or prevalence of this raccoon roundworm documented in different states. Now there is a problem here. One, nobody does this kind of field work anymore. That's one of the issues. So this is like published in 2013, but the data comes from really old, haphazard, sporadic surveys done, right? And it brings us back to the topic of like field work, right? This is not in fashion anymore. So we don't have the data. Also, the way this surveillance happens, there is a big problem there. And that's what I'm going to focus on in the next two slides. I know I need to hurry, but so we did a study in Missouri and Arkansas where we collected roadkill killed raccoons or raccoons from trappers or raccoons from another unrelated study that the agencies were doing. So we got a lot of raccoons and we necropsied them, opened up their intestines and just counted all the roundworms they had. This is the best way to have a better idea of the worms rather than doing fecal flotation, right? So we did find a high prevalence of Baileys ascaris, procyonis in the two states, about 44%. So prevalence is number that have the worms upon the total sample, right? So 44%. Now there is a problem with this prevalence, reporting this prevalence. And I want to see if I can point it out here. So we also counted the number of worms per raccoon. And this is what we got. This is a fairly typical negative binomial distribution. Uh, this is kind of like a fundamental principle in parasitology or even disease ecology. It's called Pareto principle. Have you heard of Pareto principle? It's also called the 80-20 rule. It's from economics, which means that 80% of the wealth is with 20% of the population, and 80% of the rest 80% of the population just has to do with 20% of the wealth. Same with parasites, same with pathogens. 80% of the transmission is actually done by a small number of hosts in the population, which are the super spreaders. And that is true with viral diseases, bacterial diseases, parasites, especially parasites. So that is exactly what we found, a negative binomial distribution. Most of our raccoons had zero or very few Bailey's ascaris, either 0, 1, 2, 3, 4, 5. Most of them are here. Very few are here. And this one particular raccoon, this one raccoon had 55 worms. I hope I have the picture here. All of these worms are from this one raccoon, right? So this is the typhoid Mary of the raccoon world, who has all of these parasites. The question is, what does this aggregated distribution mean for surveillance? Keep in mind, and this probably, is a thing. When I say such things, agency folks hate me. But what this actually means is the surveillance that we do routinely is inefficient because it does not account for aggregated distribution. We assume the parasite is distributed randomly in the population. Let's go and collect 20 raccoons, count the parasites and say, okay, this is the mean intensity of B pro in this population. Underlying assumption is that parasites are distributed randomly, but we know for sure they are not. Right? How does that affect our surveillance? Again, the only thing I can do is create a model, and that's what I did. So we created again a hypothetical landscape, a model which has raccoons as individuals simulated in this landscape. But more importantly, we incorporated all those processes, especially, I'm not sure if you can read it, but there is this slider here. It's called the dispersion parameter K. This K defines the aggregation. Lower the value of K, you have a high aggregation, right? So instead of 80-20, it could be 95-5. And as the dispersion parameter value increases, you will then have like a Poisson distribution, or then you can get into maybe a random distribution. But we are interested in the aggregated distribution. So we created a simple model, a hypothetical landscape, but the results are really interesting. That's what I think. So we simulated all of this aggregated distributions and then did a surveillance as we would do in the real world, right? And what it showed, and I'm just going to rush through some of these slides, but what it shows is if our inferences are based on small sample sizes, we are way off the mark, right? So the mean intensity here, so this is the real mean intensity. We know this because we have put that in the model. We are the god of this model. We can exactly say what we want and create that. But when we surveil the model population, much of our results are kind of an underestimate of the mean intensity. We are not able to catch that real value. So smaller sample sizes do not give you the real metric. Also the same with prevalence. You have a lot of variability with your prevalence result. So there is no accuracy if your sample sizes are small. And in the real world, all of the map that you saw, all of those studies are based on extremely small sample sizes. So can we really trust those? No, Florida, they say, has like medium prevalence, no, we don't know. It doesn't really mean anything. Be pro are everywhere, raccoons are everywhere, and therefore the risk is everywhere. We haven't quantified it, but at some point we will have to do that, right? And uh, we also were able to contrast interventions. What can we do to mitigate the risk of Be Pro? So very quickly, what it means is, you see the red spots, red circles here, right? These red circles are deworming the raccoons, give them dewormers to remove the Be Pro. And on the x-axis, I have different levels of coverage. So only 10% of the raccoons are dewormed, 20, 30, 50, all the way up to 90. Uh, in summary, what this says is, well, deworming can bring down the prevalence, but just from 40% to 30%. That's the prevalence, number of raccoons that have BPRO, right? On the other hand, culling of raccoons actually does not make any difference. You just add variability here. And it's might not be intuitive looking at this plot, but keep in mind, prevalence is number infected on the total population. When you cull raccoons, you are actually removing raccoons, and most of the raccoons do not have the worm. So what you are actually doing is you are reducing the total population. So you might actually increase the prevalence by culling the raccoons, because the infected raccoons are in a minority. And it's not, you cannot identify them. You just go in and remove 20 raccoons. All of the 20 could have no BPRO or very few BPRO, right? So you could actually increase the prevalence. And similarly, we wanted the interventions to be really uh, meaningful. So what we then asked is, can we, instead of intensity and prevalence, can we actually count the number of patent worms in the raccoons in the model? So patent worms, so just to give you a background, there are male Bailey's ascaris and female Bailey's ascaris. Within a raccoon, unless and until a male worm is next to a female worm, there will be no eggs, right? So if a raccoon has 5 BPRO and all of them are males or all of them are females, there is not going to be any patent female, and so there are not going to be any eggs. So we are really interested in removing the patent females, right? So how do these interventions, deworming and culling, how do they affect the number of patent worms in the population? And what you see is, deworming actually is not that effective. You bring down the number from 1,000 to 600, but culling is much more effective. If you do a very high level of culling, you can actually bring down the number of patent worms in the area significantly. But then it brings us back to the original point. Can you do this in the real world? Can you, in the real world, go and remove 90% of the raccoon population from an area? You probably could do it in St. Louis Zoo. They have a huge raccoon problem. But that's like a 5 square mile, whatever area. You can work something there. In the real world, the best you can manage is maybe 10 or 20 or 30%. So does that really work? That is something we can assess and evaluate with such virtual explorations. So finally, I think I'm cutting it very close, but this is really something I'm super interested. We are currently working on this issue, which is SARS-CoV-2, which started in 2019. Pretty soon, it kind of, the spillover happened in many animal species, especially in deer. It kind of, deer are susceptible, but they do not get the disease. So they kind of get the SARS-CoV-2, they remain infected for a few days and they recover, they have an antibody response, but no clinical signs, right? The worry is, can the deer be a reservoir of SARS-CoV-2 variants for the human population? Because what we have seen is, when the virus circulates, the strains circulate in humans, there is a very uh, huge interface, human-deer interface, and this spillover is pretty common, and deer are susceptible. Deer, once they get it, there is onward transmission in the deer population. There often is adaptive evolution of the SARS-CoV-2 in deer, resulting into variants that can be of concern for human population, and they can spill back into the human population. All of this has been documented. The question is, how does this happen? What can we do? And so again, with that in mind, um, I already had a really sophisticated model developed for the wildlife agencies in North America for chronic wasting disease. It's a white-tailed deer model with all of this, the same features. So we adapted this model for COVID. The main differences here are the CWD is caused by a prion, which is protein. But here now we have a virus. Because it's a protein, there is no adaptive immunity in CWD, but now we have this neutralizing antibody response. And more importantly, this is a prolonged course and invariably fatal. Deer don't die. They just have a short, duration of this. So this is the COVID model. What you see is the disease spreading in a deer population of a Michigan county, right? What is important here, what I really want to highlight is with this, we can generate these trajectories of the disease spread in the population. On the left, what you see is a chronic wasting disease trajectory, wherein for a long time after introduction, for about 10, 12 years, the prevalence is very low in the population and then it increases. But with COVID, because of the features, you kind of get these peaks immediately, right? And the model kind of captures that. But more importantly, and this is where I'm going to stop, is what we have shown with this model is the the spillover from human to white-tailed deer population in the region. The timing is the key. Based on the timing, the population pattern, the transmission in the deer population will change. And the reason for that is you need to understand. So you look at introduction in August, September, October, November, or June and July. That results into like an outbreak that kind of is stamped out immediately. Unlike introduction in December to April, you have a multi-year outbreak. So you have like long transmission chains, right? The reason for this is the sociality of white-tailed deer. As I said, there are those social groups, there are bug groups, and their contact pattern changes seasonally. And that is incorporated in the model. And therefore, we are able to capture this nuanced, changes in the pattern of COVID. More importantly, this can help us understand the transmission chains, the evolutionary dynamics of SARS-CoV-2, and we are trying to use this to inform wildlife agencies for surveillance of COVID. World Health Organization has recommended that we undertake this surveillance everywhere, especially because of variants of concerns. And to do that, it's not just any samples, but can we combine wastewater surveillance for a region with the model explorations to inform agencies when to sample and when to look for VOCs and when maybe not to sample. So that is the goal of this work, and hopefully it will be out soon. I just want to end up with this note that modeling approach I talk about, the virtual explorations, does not replace field work or field experimentation. It complements it. We actually work with the limited data that is available and kind of use it in a formal framework. It helps us better understand these complex systems. And it's a great alternative to direct field tests, which often are not possible, and support defensible decision making. That's all I have for you today. Thank you so much. Any questions? Yeah.
Yeah, thank you so much, Ani. Raise your hands and I'll bring the microphone to you.
So I was just curious, given the difficulty in doing enough field sampling, whether the sort of emerging eDNA techniques would really, sort of the counterpart to COVID in wastewater is in streams and rivers. And I was wondering about other wildlife diseases and whether you see a sort of new emerging field of eDNA substituting for actual species sampling.
It definitely is going to be it is something that needs to be integrated in this sampling surveillance framework. And we actually have a new collaboration with University of Minnesota, where they have developed these new techniques very similar to eDNA. But in this case, they have this new technology that can be applied to detect prions in the environment, right? So that has changed the way we do surveillance for CWD. But it's something that needs again to be systematically implemented in the real world. And for that, we need a formal framework like this to understand how do we use these new technologies. So that is a great question, suggestion. And eDNA, of course, is going to revolutionize some of the surveillance we do for diseases, maybe chytridiomycosis, something like this that is, that we are struggling with. But yes, these emergent technologies are going to change the way we can kind of deal with these disease systems.
Anyone else? Yeah.
Do the deer cough in the face of people? Is that how they get COVID from deer?
I have seen not studies but observational reports wherein they believe that the way deer get COVID is from disposed of masks in areas where people go out for hiking and all of. But again, that's not no citation there. But there is a huge interface where people feed deer in their backyards, even if it is not allowed. Deer often come to the garden, and then go to YouTube, there are videos where there is this woman going to her backyard and calling for a deer, and then the deer will come and eat from her hand. So these interfaces are not all really maybe legal or allowed, but they exist. And because we have like 30 million white-tailed deer in US, and urban, rural, suburban, everywhere. So I think that interface that is what makes white-tailed deer really problematic for a disease like this. And I just did not talk about it, but you know about this Delta variant. There was a time when we had Alpha variant circulating in the human population, and that was displaced by the Delta variant. Alpha went out of circulation from humans, but it persisted in deer population, and it evolved in the deer population. And then it escaped the immunity provided by the vaccine to humans, and there was a case of human infection. Thankfully, we don't have documented any more cases of such thing, but who knows? If that remains a persistent source, then that is something we will have to deal with. I don't want to fear monger or say that, but it's alarming. Yes.
Yeah.
Yeah. This end. Perfect. Amazing talk. So cool. The modeling of nonlinear systems into hypothetical landscapes is so cool. It's what Jurassic Park was based off of, and it's what the first supercomputers were made for.
Right.
And I love Jurassic Park. So the question is just these models that you have, all the factors that are listed out, very cool, like the adjustable sliding scales. With the non-linearness of these things, what is your confidence that they are being fully captured? Because there's so many things that can't be captured, and that's why the dinosaurs escaped. Like these mask stories, and the food stress is a thing for the birds. Is it a thing for the raccoons? Is it a thing for the deer? That's what I'm getting at.
Now, you are the first one to ask this question in exactly the right wordings, I would say. So I work with wildlife agencies, and they always, the same question they ask me in a simple way: how do we trust these models? And my answer to them, I'll give you a more elaborate answer. But to them, my answer is, I am not asking you to trust these models. I'm asking you to use these models, parameterize them the way you think the system works. And so it essentially is a what if scenario analysis. I always make it a point to say that we are not predicting anything. These are not predictive models. I don't believe any model of a complex system can predict what's going to happen. We did that mistake in 2020 when some of the models for SARS-CoV-2 were described as predicting something, and they failed, it was not the failure of the model, it was the failure of people to comply with the basic, requirements. So keep that aside. These are not predictive. You don't have to trust them. You have to use them in the sense like design these what if scenarios. So with your question, let's say about the trajectory for the COVID in white-tailed deer, right? Uh, we can actually compare and contrast several interventions there, right? And it actually will help us better understand the mechanism of the system, not necessarily the what do you call it? Like, the effect size is not important, but the difference between the results is important. So if we do X, Y goes down. If we do Z, Y goes down significantly, right? So X and Z, there is a difference. I would prefer doing Z because based on the current best understanding, we have a better outcome from the model. And this is all adaptive management. So if we have new data available, we can rerun the model and update our thing. So I think it should be incorporated. This virtual experimentation should be incorporated with policy and decision making, rather than expecting it to guide you, give you an answer like a crystal ball. It should be incorporated in the policy decision making, wherein you put in the best information, get your results, and you make decisions based on these results. So defensible decision making. Your data improves, things change, rerun the models, bring back the new results, compare them with the old results, and justify why we are changing the strategy. I think that is a tool for decision making, for supporting the decisions, rather than a tool which tells you this is what it is going to be. Does that make sense?
Yeah.
Right. So I think it's more of a tool than like, the agency folks always want an answer, and which no models can provide, because they always come with this is based on all of this being true. And we have no control on many of those things in the real world. So I think, if you know of sensitivity analysis, that's where the answer lies in technical terms. So I don't trust any of this, but I use a parameter that I don't know the value of. I don't know whether what I'm using is correct or wrong, but then I run the model with a range of values for that parameter. And doing this analysis, it then tells me that if the value is above 80, it's going to make a huge difference. But if the value is between 0 and 80, it doesn't change anything. So then it leads me to focus on that parameter, go to the field, collect new data, make sure that I prioritize that particular aspect of the system, and come back and then run the model. So it's an ongoing process, not necessarily a plug and play and a model that will give an answer. Almost always raises more questions every time you use it in the right way. Yes.
Yeah, sure.
I'm curious if there's room in these agent-based models to apply certain aspects like mutation rate and selective pressures to, I guess, track evolution of variants from the pathogen, but also adaptive responses of the hosts.
That is exactly what we are trying to do with Jana. So, I think a breakthrough for us was to be able to quantify the transmission chains. So now with the current model of SARS-CoV-2, we are able to actually count the number of continuous transmissions occurring in the population. And now the person who has the expertise with the adaptive evolution, is trying to match it up with the mutation rates and the generation times and the probability of emergence of certain traits. So there are two ways of doing that. That can be either done in the model, or it can be done using the transmission chains outside the model. And we are trying to figure out what would work best. But that's where we want to go. And I think there is a lot of room to do that. And others have done that for other systems.
Let's take one last question. So there's a question online, and Amartya asks, is there anything special about white-tailed deer that we want to study SARS-CoV-2 in that, or are there other animals that are other important hosts?
Right.
So-
The answer to that question is that there are several other animals wherein who are susceptible to SARS-CoV-2 and have ongoing transmission once the spillover happens. But deer pose a special problem in North America because of their numbers and their ubiquity. Right? Also, the deer-human interface is really dynamic in North America. These are harvested every year, so hunters come in contact with their secretions and excretions and body parts, 1 or 2 months every year, which is a huge thing in terms of pathogen transmission, right? And in case these deer turn up to be a novel reservoir for uh, uh, for the SARS-CoV-2, there is a high likelihood that we will see more variants because of the sheer number of transmissions that can happen in the pop. The longer the transmission chain, the higher the probability of VOCs arising. And that is the reason why deer are really important in North America. There are other mink farms. They have shown to have these transmissions and even some of these mutations, but deer are special for that reason. Yeah.
Well, thank you so much, Ani.
Thank you.